Hello everyone, it's Sindhushri from Molecular Biology Department. Myself and Kavya Shri will be talking about the enzyme ligase. These are the contents which includes introduction, the enzyme ligase, nomenclature and classification. We'll be mainly talking about DNA ligase, history, types, enzymatic mechanism, application, conclusion and references. Cutting and pasting are two of the first skills children learn, and the tools they use are scissors and glue. Similarly, cutting DNA and pasting DNA fragments together typically are among the first techniques learned in the molecular biology lab and are fundamental to all recombinant DNA work. Such manipulations of DNA are conducted by a toolkit of enzymes. They are restriction enzymes, polymerase enzymes, DNA ligase, host cell, and vector. Restriction enzymes are used as molecular scissors, whereas DNA ligase function to bond pieces of DNA together, and a variety of additional enzymes that modify DNA are used to facilitate the process. Thus, restriction enzymes and DNA ligases represent the cutting and joining functions in DNA manipulation. All other enzymes involved in genetic engineering fall under the broad category of enzymes known as DNA modifying enzymes. These enzymes are involved in the degradation, synthesis, and alteration of nucleic acids. The term ligase is derived from a Latin word ligare, to means to bind. Ligases are also called as synthetases. They catalyze the joining of two large molecules through the formation of new chemical bonds such as carbon oxygen, carbon sulfur, or carbon nitrogen with the hydrolysis of ATP. The general reaction is we have two reactant A and B in the presence of the enzyme ligase. By utilizing ATP, it catalyzes the formation of AB complex. The nomenclature. The common names of ligases often contain the word ligase, like DNA ligase, a frequently used enzyme in molecular biolaboratory. Synthetases is another commonly adapted name for ligases since they are applied in the synthesis of new molecules. Biochemical nomenclature has sometimes distinguished synthetases from synthesis and sometimes treated the word as synonyms. Synthase is a lyase that catalyzes the breaking of various chemical bonds by means other than hydrolysis and oxidation and does not require any energy, whereas a synthetase is a ligase that binds two molecules and thus require energy. Coming to classification, ligases are classified as EC6, that is enzyme commission number 6 in the EC number classification of enzymes. Ligases can be further classified into 6 subclasses. EC 6.1 includes ligases used to form carbon oxygen bonds. In EC 6.2 includes ligases used to form carbon sulfur bonds. 6.3 includes ligases used to form carbon nitrogen bonds. 6.4 form carbon carbon bonds. 6.5 forms phosphoric ester bonds. 6.6 includes ligases used to form firm nitrogen metal bonds. In a routine day, we come across so many adverse conditions such as radiation, high heat, chemicals, and mutagens. Those agents mutate our DNA or break it, but our body's natural repair mechanism repairs our damaged DNA, thus, we remain safe or unmutated. Many enzymes are involved in doing such DNA repair and replication. One of the such enzymes are DNA ligase. Exact cutting of DNA molecule is very useful for DNA cloning. Its full potential is only exhibited when the fragments produced are joined together to give a new structure known as recombinant DNA. The most common DNA ligase is isolated from the bacterial virus, that is T4 bacteriophage. DNA ligase catalyzes the formation of phosphodiester bond between two deoxynucleotide residues of two DNA strands. DNA ligase enzyme requires a free hydroxyl group at the 3' end of one DNA chain and a phosphate group at the 5' end of other, and requires energy in the process. E. coli and other bacterial DNA ligase utilizes NAD+ as an energy donor, whereas in T4 bacteriophage, T4 DNA ligase uses ATP as cofactor. Coming to history, the first DNA ligase was purified and characterized in 1967 by the Gillard, Lehman, Richardson and Horwitz laboratories. The common commercially available DNA ligases were originally discovered in T4 bacteriophage, E. coli and other bacteria.
Hey everyone, I'm Kavya. So from here, I'll take on. I'll be talking about the different types of DNA ligases. So I'll be talking about the three here. So the first one is the E. coli ligase. The E. coli DNA ligase is encoded by the like gene. The DNA ligase in E. coli, as well as most prokaryotes, uses energy gained by cleaving the NAD to create the phosphodiester bond. It does not ligate the blunt-ended DNA except under conditions of molecular crowding with polyethylene glycol and cannot join RNA to DNA efficiently. The activity of E. coli DNA ligase can be enhanced by DNA polymerase at right concentrations. Next up, we have the T4 DNA ligase, that is the ligase from the bacteriophage T4. So, this bacteriophage infects the E. coli. The T4 ligase is the most commonly used in laboratory research. It can ligate either cohesive or blunt ends of DNA, oligonucleotides, as well as RNA and RNA DNA hybrids, but it cannot ligate the single stranded nucleic acid. It can also ligate the blunt ended DNA with much greater efficiency than E. coli DNA ligase. Unlike E. coli DNA ligase, the T4 DNA ligase cannot utilize NAD and it has an absolute requirement for ATP as a cofactor. Next, we have the mammalian ligase. In mammals, there are four specific types of ligase. The first one is the DNA ligase 1. It ligates the nascent DNA of the lagging strand after the ribonuclease H has removed the RNA primer from the Okazaki fragments. Next we have the DNA ligase 3. Complexes with DNA repair protein, that is the XRCC1, to aid in sealing DNA during the process of nucleotide excision repair and recombinant fragments. Of all the known mammalian DNA ligases, only ligase 3 has been found to be present in the mitochondria. Next up, we have the DNA ligase 4. So, this DNA ligase 4 complexes with XRCC4. It catalyzes the final step in the non homologous end joining, which is one of the DNA double strand break repair pathway. The DNA ligase 4 is also required for a VJ recombination, the process that generates diversity in immunoglobulin and T cell receptor loci during immune system development. Next, we have the DNA ligase 2. So, this DNA ligase 2 appears to be used in repair. It is formed by alternative splicing of a proteolytic fragment of DNA ligase 3 and does not have its own gene. Therefore, it is often considered to be virtually identical to DNA ligase 3. The DNA ligase from eukaryotes and some microbes uses ATP rather than NAD. So next up we'll be talking about the enzymatic mechanism of DNA ligase. So as you can see, this will happen in 4 steps. So this image will demonstrate how ligase will catalyze two DNA fragment strands. Okay, so the thing that is present in yellow color, which is an oval shape, that represents the ligase. So the enzymatic mechanism of DNA ligase is to form two covalent phosphodiester bonds between the 3' hydroxyl end of one nucleotide, which acts as an acceptor, with the 5' phosphate end of another, which acts as a donor. So two ATP molecules are consumed for each phosphodiester bond formed. Okay, AMP is required for the ligase reaction, which takes place in 4 steps. So in step 1, what happens is that there will be reorganization of activity sites such as nicks in DNA segments or Okazaki fragments. In the second step, there will be adenylation of lysine residue in the active center of the enzyme. So adenylation means there will be addition of AMP and pyrophosphate will be released. And in the third step, there will be transfer of the AMP to the 5' phosphate of the so-called donor, and there will be formation of a pyrophosphate bond. Okay, in the fourth step, what happens is that there will be formation of a phosphodiester bond between the 5' phosphate of the donor and 3' hydroxyl of the acceptor. So ligase will also work with blunt ends, although higher enzyme concentrations and different reaction conditions are required. Okay, next we'll move on to the applications of DNA ligase. DNA ligase enzymes play an important role in genetic engineering. In the absence of DNA ligases, recombinant DNA technology cannot be successful. The important functions of DNA ligase enzymes are as follows. So the first application is genetic engineering experiments involve joining of DNA fragments to produce recombinant DNA molecules. Ligase enzymes are used in joining process. As we all know, ligase's job is to glue things, right? Uh, the second application is ligase enzymes helps in ligation of vector and inserting recombinant DNA. The third application is they help in ligation of linkers or adapter molecules at the blunt ends of DNA fragments. They help in sealing nick in double-stranded DNA. So the next step is that ligase enzymes require 3' hydroxyl and 5' phosphate groups for ligation. So next, moving on to the conclusion. So I'll be talking about how the ligase can be used as a drug target. In recent studies, human DNA ligase 1 was used in computer aided drug design to identify DNA ligase inhibitors as possible therapeutic agents to treat cancer. Since excessive cell growth is the hallmark of cancer development, targeted chemotherapy that disrupts the functioning of DNA ligase can impede adjuvant cancer forms. Furthermore, it has been shown that DNA ligases can be broadly divided into two categories, namely ATP and NAD plus dependent. Previous research has shown that although NAD plus dependent DNA ligases have been discovered in sporadic cellular or viral niches outside the bacterial domain of life, there is no instance in which a NAD plus dependent ligase is present in a eukaryotic organism. The presence solely in non eukaryotic organisms. Unique substrate specificity and a distinctive domain structure of NAD plus dependent compared with ATP dependent human DNA ligases together make NAD plus dependent ligases ideal targets for the development of new antibacterial drugs. These are our references. We thank the Department of Molecular Biology for providing us the opportunity to give this little presentation. Thank you.